Ouch. The endomembrane system is a system inside the cell that regulates the traffic, specifically protein traffic. That's where we're going. It also does some metabolic functions, and you'll see these players. We've already talked about the nucleus. The ER is right next to the nucleus. You'll see the Golgi apparatus that's there, sometimes called the Golgi body. You also see things called vacuoles, lysosomes, and then it all ends with the cell membrane. An important thing to remember is that these are all continuously kind of connected. They're just membranes that are right next to each other, and that's pretty exciting. Obviously, here you can see these guys here. You can see the nucleus. You can see the nucleus, you can see the ER, you can see the Golgi connected. Here's a lysosome guy right here. His job would be to do something in that process. This isn't a plant cell, so we don't see any vacuoles, but the important thing is then there's the membrane. There's really no that is wasted. They're all right next to each other, and they all have a reason for being there. Make sure that you know these objectives by the end of 6.4. Let's talk about the endoplasmic reticulum first. You'll see that endoplasmic reticulum is both smooth and rough, and the reason why it's called rough is when the first guys and gals who had the microscopes to be able to look at it that well, they saw this thing that they named the endoplasmic reticulum, and it had little balls on it, and you can see here in the picture, and they called it rough. It just looked rougher. There was another portion of endoplasmic reticulum next to the nucleus that was actually it was actually smoother. So we now know that smooth ER lacks ribosomes, and those little wheels on the bus, or the balls, or the dots on the rough ER are actually ribosomes. So you probably kind of have an idea already from 6.3 what rough ER is going to do. But let's talk about smooth first. You can see the four things down there in the bottom left corner that smooth endoplasmic reticulum does. Smooth ER creates lipids. You can see also that it does a few other things. Make sure that you know those four things that smooth ER does. Rough ER, as we already talked about, helps make proteins. It's where translation takes place. It also is a membrane factory for the cell, and it also kind of packages those newly created amino acid strains, which we'll call polypeptides, into vesicles. Again, it's just phospholipid little packets, and sends them away to the Golgi apparatus. Make sure you know those four things that the rough endoplasmic reticulum. Over here you'll see this is the receiving side of the Golgi apparatus, where I'm kind of moving the cursor. You'll see a bag coming in there with maybe an amino acid chain, and that's coming from the rough endoplasmic reticulum, and it's going to go into the Golgi. The Golgi will then do something to that amino acid chain, and it will modify it. A lot of times it can take, the Golgi is incredibly, in my opinion, intelligent, although it doesn't have a brain. It can take a singular sequence of amino acids, which has come from a singular sequence of DNA, and it can modify it. It can modify it. It could add a carbohydrate here, or it could add a phosphate group here, or it could twist it in a certain way here, or it could remove an amino acid, and the whole thing folds differently. The point is that the Golgi can make many different functioning proteins from just maybe in theory one original amino acid strand, which came from DNA. It's amazing how the Golgi can kind of expound upon a singular linear message. It can turn it into 20, 30, 50 different things if the cell needs those things. The important point here, as far as the structure goes, is that it has a cis side and a trans side. We'll get in later to what cis and trans means, but just understand that C comes before T in the alphabet. That's how I remember that the cis side is the receiving side, and the trans side is the side where the then modified amino acid chains then can leave. These amino acid chains will leave in a phospholipid covering, a vesicle, if you will, just like the endoplasmic reticulum, and then those will go then to the membrane. And we'll get to the plasma membrane later. Make sure you see these and memorize these three big points about the Golgi apparatus. It's a really cool thing, and if you know the Fish song Golgi apparatus, then you know it. Please press pause. I'll be asking you tomorrow what the answer is to this question about the Golgi apparatus. Here's kind of the big picture, and this is figure 616. You can see here the rough endoplasmic reticulum. You maybe see something budding off here. Maybe there's some amino acids put inside of a vesicle. It's then going to go to the cis sides of the Golgi. It'll be further modified within the Golgi apparatus. And then on the trans side, it's going to be packaged in a vesicle again and leak out and go all the way, sorry I pointed at the screen, to the plasma membrane. You see that there. Then the package's components will then be flushed out. It may be into the bloodstream. It may be go somewhere. Maybe it's a hormone like testosterone produced in a cell of a testicle in a man, and now this protein hormone is being pushed into the bloodstream to maybe go up to a young man's face, so that now he starts growing facial hair or something like that, to give you some context. Another component, cellular organelle of the endomembrane system, are lysosomes, and these guys I think of as digesting centers. They have these hydrolytic enzymes, and you know by breaking down the word hydrolytic, it means that it's using water to cut. So these guys use water to cut, and there's water within them, but they also have a lot of protons floating around, these H+ ions, and these H+ ions create a very acidic environment in there, and that's why I have danger acid. You have within every one of your cells these little bags called lysosomes that have a pH of around 2. It's like battery acid. I mean, if you put your finger in a lysosome, if you had a giant lysosome as big as a basketball, your finger would be digested down to the bone within maybe 60 seconds. That's how powerful these things are. They're also looked at as trash cans. It's where all the waste inside the cell goes, and it's where everything is broken down, and the components, the little nucleotides or the individual amino acids or atoms, then can be reused by the cell. And you can see down here that the lysosomal enzymes can hydrolyze all these things. Here are some examples of lysosomes in actions. This is phagocytosis. It's where a cell actually, their outer membrane actually engulfs something. Let's say it's a food molecule. Let's say that we are in, let's go easier. Let's say that we are a bacteria, and a bacteria is going to engulf something out in its environment, and actually the membrane will move, and you can see that here. And once that vesicle of something comes into the cell, the lysosome can then bind to it, and it can digest the components. And this is called phagocytosis. You can see then the literature I've put in the bottom right-hand corner of this. Here's an example of a white blood cell that's going to phagocytize or bring in, these look like bacteria, cocci bacteria that might be in your digestive tract that aren't yours. And this guy's grabbing them, pulling them in, and he will then combine these bacteria with a lysosome within him to digest them so that they can't harm you. Also, there's this process called autophagy, and this is to recycle the cell's organelles. We talked about this earlier. Maybe we have an old mitochondria here. Its days have gone. It used to create energy for the organism, and now it's old. This is a part of everyday cellular life, that the lysosome will then come in and digest organelles that are old, and you can see how that happens here. Finally, last part of the endomembrane system in 6.4 are vacuoles. And vacuoles are found in plant cells. And vacuoles' main function is to store water, but they also store lots of metabolites, organic compounds, things that the cell is going to use. They serve to help detoxify poisons that may come into the cell. They store calcium. They do a whole bunch of stuff. It's just a big bag of water, and you can see here it's pretty prominent, and it's even centralized. It says central vacuole here in a plant cell, and it's going to help the cell maintain water balance. That's it. Make sure that you answer this question, please, and then spend some time answering these questions, these objectives, and we'll talk about them tomorrow. Make sure that you know these. Thanks for your attention.